On our lesson for today, we are going to describe the structure, nature, and properties of DNA molecule. Welcome to Teacher Gia's channel. I hope you've already gone through my last lesson, which was talking about describing the structure, nature, and properties of chromosomes. So let's begin. Structure of DNA. DNA, also in full deoxyribonucleic acid, is a double helix structure composed of two complementary strands. It was first described by Watson and Crick in 1953. The two strands run antiparallel and are held together by hydrogen bonds between complementary nitrogenous bases. We move to components of DNA. We have the first one, which is nucleotides. DNA is made up of repeating units called nucleotides. These are the building blocks of DNA, each comprising a phosphate molecule or group, a deoxyribose, which is a 5-carbon sugar, a nitrogenous base, 4 types available, that is adenine, thymine, cytosine, guanine, and guanine. The initials are A, G, C, and T. The sugar alternates with the phosphate, and the two form the backbone of the strands. We've said the two components of DNA are the phosphate molecule and deoxyribose, which is a 5-carbon sugar. So when they alternate, they form the backbone of the strands. The bases, which are the adenine, thymine, cytosine, and guanine, combine in a specific manner such that adenine pairs with thymine and guanine pairs with cytosine. The bases are held together by hydrogen bonds. So when you're asked to name components of DNA, you will start with the first one, nucleotides, then the second one, sugar phosphate backbone. I hope we've talked about that. And then the third one, you write a nitrogenous base or base pairing, which means that the adenine pairs with the thymine and the cytosine pairs with the guanine. So kindly read this part that I've written here and understand. We'll move to functions of DNA. Genetic information storage. Encodes instructions for protein synthesis. Another function of DNA is replication. Can duplicate itself to pass genetic information to new cells. And lastly, we have protein synthesis. The sequence of bases that make up a gene determines the sequence or arrangement of amino acids to make a particular protein. The proteins formed are used to make cellular structures, enzymes, and hormones. The types of proteins an organism manufactures determines its characteristics. For instance, albinism results from a failure of the cells of an organism to synthesize tyrosine, an enzyme essential for melanin production. And lastly, we're going to talk about properties of DNA. It's not the last one. I think it's the second last because we're going to also see the differences between DNA and RNA. So properties of DNA. The first one, you have replication. DNA can make identical copies of itself during cell division, have storage of genetic information, encodes the instructions for protein synthesis. It serves also as a function. Mutability. DNA can undergo mutations leading to genetic variations. We have transcription template. It acts as a template for mRNA synthesis during transcription. And we have stability. Double-stranded structure or nature provides stability, protecting genetic information or code. So the last part that we're going to talk about today is differentiate between RNA and DNA. So we have one column for feature DNA and RNA. So we have full name for DNA is deoxyribonucleic acid, while RNA is ribonucleic acid. We have structure. DNA are double-stranded, RNA are single-stranded. Sugar component, DNA is deoxyribose while RNA is ribose. Nitrogenous bases in DNA are adenine, thymine, cytosine, and guanine, while nitrogenous bases in RNA are adenine, uracil, cytosine, and guanine. The base pairing in DNA, A pairs with T, that is adenine pairs with thymine, and cytosine pairs with guanine. While in RNA, adenine pairs with uracil and cytosine pairs with guanine. Functions. The function of DNA is it's a long-term storage of genetic information, while in RNA, it's a temporary transfer of genetic information for protein synthesis. We have types. DNA 1 is a one main type. There's one main type, while RNA 3 types, mRNA, tRNA, and rRNA. Location. DNA is confined to the nucleus in eukaryotes. RNA is found in both the nucleus and cytoplasm. And lastly, we're going to talk about stability. DNA are stable while RNA are less stable. So that is the end of our topic for today. And in the next lesson, we're going to distinguish between F1 and F2 generation. See you. Bye and take care. Don't forget to subscribe.